Some of them give me a different direction and dimension of how to interact with it. So like neuroscience, I can kind of play on the electrical side. On in regenerative medicine, we can play on the mechanical side. With cancer, we can play with the photonic side and the energetic side of physics. And this is kind of where my PhD grew from, which where I saw that magnetic fields can alter cancer physiology. And I kind of flipped that question. If cancer can be interacted with magnetic fields, what is it emitting? And can we use that as any kind of diagnostic? So that's a paper that we published to show very specifically that launched this entire biophysics world for me to show that when you stress cancer cells, when you change their energetic landscape, they have a optical fingerprint. They have a light fingerprint that we can differentiate between healthy tissues. So we've shown this in cancer cells in dishes. We've done this in mice that we've injected with different kinds of cancers and used it as a platform, used these light signals to say, is there cancer in the animal or not? And what we're hoping to do is in humans, and that's what we're working on right now, is use these optical sensors to see if we can classify whether a patient has cancer or how far the cancer is in terms of its growth, non-invasively from the outside of their body.
Wow. So if I'm hearing you right, what you're basically saying is, okay, obviously cancer cells are different than non-cancer cells. They became different. They have a different type of metabolism. And to the extent that, we said that these biophotons get produced from based on metabolism that's happening, the cancer cells are metabolically distinct from the non-cancer cells, and therefore you would expect them to have a different photonic signature that they emit. And what you're saying, I think, is that you could basically detect cancer cells because they have this unique light signature they're emitting, and you can do this in a fairly non-invasive way.
Absolutely. That's exactly what we're doing. It's taken a lot of data points, obviously, because there's heterogeneity between cancer that we're starting to see. I mean, starting to see it's well established now, but yes, you can tell the difference between what is cancerous and what is not cancerous. What we're having a hard time doing right now is telling the difference between the cancers. If we're
Mm-hmm,
looking at
mm-hmm.
melanoma versus breast versus glioblastoma, what is that optical signature? So this is where we're looking at more of the physical dimension of the light, so the wavelength, the intensity, the frequency patterns, what is different about them? And this is where we're kind of leveraging AI to help us figure out what those pattern differences are.
Mm-hmm, mm-hmm. And I think going back quite a long time, people have had ideas about what cancer is and how it arises. Obviously, one of the classic textbook ways people think about this is what people call the somatic mutation theory. You get certain mutations and oncogenes, and maybe that causes cancer. I've had other people on the podcast that have a very different view. People like Thomas Seyfried, who say, "Well, no, this is cancer is mostly a metabolic dysfunction thing, and it's not so much a somatic mutation thing." And obviously, when we start thinking about things like light, um, people always associate UV light as causing mutations that causes cancer. How do you,
Mhm.
as a biophysicist, think about what cancer is and how it maybe ties into concepts from the regeneration field? Do you view this as primarily a
Mhm.
mutation thing, as a metabolism thing? How do you start to wrap your head around it?
Absolutely. And I'm actually kind of working on a paper on this to help solidify this idea from an energetics point of view. And I view it as a dysfunction in energy in terms of how the energy is regulated and how the energy is dissipated. And the mutations that arise might become because of shifts in wavelengths and the energy metabolism of the cell itself. We know that from our papers that there's a shift in wavelength from blue to red, and you can kind of fingerprint those. And the more you lean towards the blue, you emit more UV photons. And if those UV photons lead to more mutations, then that accumulation will change the energy dynamics within the cell. And then the cell will need to adapt. So that's where the free energy principle comes in. The cells will always do the behavior that it requires the least amount of energy for the system. So there's a different multi-layered dimension here is that I believe that it's fundamentally energetics. When there is changes in energetics, the physics match that to produce a behavioral response that the cell can adapt to, and which is what we see as highly proliferative and highly plastic. And you brought an excellent point there about plasticity, about stem cells and cancer. why both of them are highly proliferative. One is very plastic, but the other one is also plastic, but very mispatterned. Where's that information loss
Yeah.
that allows something to become patterned versus not? And I think that's really what I want to get into is information. They're both energetics. They're both functioning at, physical and chemical landscape, but where's the mispatterning happening?
Mhm. Say a little bit more about, so what did you say around cancer cells, being more or less red versus blue shifted? What exactly did you mean there?
So the photon emissions that are being emitted
Yeah,
from cancer cells are more blue shifted than their healthy counterparts.
I
So
see.
we've seen this within melanocytes and melanoma, is where we've spent most of our time on. The melanoma cells and the tumors that are forming within rodents were more blue shifted than they were red shifted than their melanocyte counterparts.
Okay, so if I'm putting some of those ideas together, some kind of energetic disturbance happens and a cancer cell forms some way, somehow. And it's now energetically different than it was before it became a cancer cell. There's this free energy principle that I think you were pointing to. And basically, what you're saying is cells are generally going to try and be energetically efficient and use no more energy than they probably want to or need to. And so maybe in some sense, a cancer cell is one in which the cell is adapting to an energetic shift to obey this free energy
Mhm.
principle, but it's disturbed in a way that makes the cell's adaptation at its own level counter to the organism's. Uh,
Correct.
yes. Okay.
Exactly. So the decisions that it makes, it anthropomizing it,
Yeah.
is independent of the collective of where it came from. And
Yeah.
so that disconnect is where we get, I think, tumors. But ultimately, it's arising from this change in energetic states, whether that be because of photonic inputs or dysregulation in photonic inputs. For example, we know that flavins and porphyrins, which are molecules that absorb red light, are altered within cancer.
Yeah.
So, is that the cause? Is that the result? Those are the gaps that we need to fill.
Yeah. Yeah. And yeah, lots of question marks, lots of exciting questions to pursue here. But yeah, obviously the role of light in biology extends way beyond image formation and way beyond even just circadian regulation. This stuff is fascinating.